Hello, everyone. I'm Dennis. I'm a PhD student at the moment at the University of Oxford in the Booth Group, and I'm working on using light to understand and control biology. So very different from solid state chemistry. We're now in the liquid phase and working in a biological environment. So before I tell you about what I have spent the last 4 years of my life doing in my PhD, I'm going to tell you a bit about myself. So I'm Dennis. I'm originally from Austria, from a small village outside of the town of Feldkirch, so right at the border to Liechtenstein in Switzerland and not too far from Germany. That's where I grew up, and that's also where I did my A-levels. I did my A-levels in chemistry, German, English, music, and maths in this beautiful building right at the river where I spent many hours not paying attention, rather just looking out of the window and admiring the scenery. But still got my levels. It was a great time there. And at that time already, I was thinking, oh, I like science. I really enjoy doing something practical. I like working things out. So I started to think, I want to study chemistry. That seems cool. I liked like working with Bunsen burners. I liked the colors. I liked mixing things. So I started thinking, oh, that might be great. And I actually had a lot of time to think about what I want to do because in Austria we still have compulsory military service. So I took a year to do that, but I refused to carry a weapon. So instead I did community work as an alternative to military service, and I was working with the diocese, and I worked for 9 months with kids and adolescents, preparing leisure time activities for them and doing workshops with them. And actually left me with a lot of time to think about what I want to do. And during that time, it really became clear that I want to do chemistry and I'd like to study abroad for that. And so I applied to go to several places actually in Switzerland, in Germany, and in the UK, and I got an offer from Oxford. And so I took that offer, but I had a few months between the end of my community work and when my degree actually started. So in between, I worked at a actually quite well-known chemical supplier, Sigma Aldrich, just across the border in Switzerland in Books, where I spent 6 months roughly handling chemicals, taking samples for analysis, and actually getting a really good grasp of chemical safety, which actually was very, very important throughout my whole time as a chemist. And then after that, I took the big trip over to the UK, to Oxford, where I started my undergrad, actually at the same time as Nicola, who we just heard. And I was at St. Anne's College for that. And I'm going to tell you just a tiny bit about the course. Nicola's already mentioned a lot, but we've got the first 3 years, a very classical degree experience with lectures in the morning and labs and tutorials in the afternoon. And then the fourth year, the final year being the MChem year, really the highlight of the course, where we spend 1 year doing actual research in a research group in a department. And I really enjoyed that, and I wanted to do more of that. And I did that in chemical synthesis, and I very much enjoy synthesis. So I applied to do a PhD in synthesis, and I joined then the Synthesis for Biology and Medicine Center for Doctoral Training, the SBM CDT, at Oxford to do my PhD. And as part of that, I joined a different college, Wolfson College, which was just up the road. And I also joined the Booth Group, where I undertook my research. And that's obviously my route to where I'm now. I'm at the end of this PhD journey. And I can tell you a bit about what comes next, but also other routes that people can explore. So obviously, you have to do A-levels if you want to go to university, or at least some equivalent to it that allows you to study at university. And from there, you can go into what I did, an integrated master's degree, and then go down the academic pipeline. Or if you're not really academically minded, if studying is not for you, or you don't know what to do, you can go into employment if you have a job you want to do, or you do the slightly more convoluted way that I had to do, which is take a gap year to figure out what you want to do in your life, and then you can go into employment, or you can go into study, or go into study via employment like I did. And then a university degree really gives you a lot of, it equips you with a lot of skills that you can bring into the public sector in employment, either the hard skills, such as chemical synthesis that is required for a lot of pharmaceutical companies, for example, but also the soft skills like the analytical thinking, the mathematical thinking, that is really valuable. But you don't have to do anything that's related to that, because a degree just shows that you're able to think and to develop skills on your own as well. And so a lot of my friends who studied with during undergrad are now doing a lot of different things that have nothing to do with chemistry, being programmers or actually having done law conversions and now solicitors. In my case, I then went on to do a PhD, which is also an option. That in itself opens more doors in the employment sector, as the skills that a PhD offers you are really highly sought after. And many people I know after a PhD go into the open sector and work in teaching or in industry. In my case, I'm going to continue the academic pipeline, and I'll start a postdoc, as Nicola already mentioned that she is doing at the moment, in about 2 to 3 weeks. So my PhD really is at the end, and I'll be working again in chemical synthesis because that's what I really enjoy. And hopefully after that, I find either another postdoc or maybe even an academic position. And so we'll find out where this will take me. But at the moment, I'm right here at this gap. Now, what I've been spending my last 4 years doing is my PhD, where I develop methods to control DNA function with light. And I want to preface this with saying that cellular biology is really, really difficult. What you can see here is 3 different cell types. We've got here a white blood cell in the top left. We've got neurons here at the bottom left, and over here we've got bone cancer cells. They could look not any more different, could not have any different function. However, they all have one thing in common, and that is that they all share the same genes and do the same process, which is called the central dogma of biology. The central dogma of biology states that DNA gets transcribed into mRNA, which then gets translated into protein. And these proteins do almost everything that we know in biology, and they give rise to biological function and also the phenotype, so how an organism presents. You might be familiar with the very classical example of pea flowers showing different colors depending on their genome, and that actually is determined by proteins. And if these flowers can actually produce a protein that produces that color depends on if they have the gene for the specific protein. But they both do very similar things because their genome is very, very similar. Now, there's a whole host of different proteins out there. They could look not any more different and have any more different function, but they're all really important. And I've just shown here at the bottom 3 that they could not look any more different. And I'm just going to briefly walk you through different types of proteins and why they're so important. Now, one class that you might already know is enzymes. Enzymes are there to break down molecules and build other molecules. So if we eat food, that gets broken down by enzymes, and then our body takes other enzymes to take the things that it has broken the food down into to make other things that are useful for us, including, for example, other proteins. Then we have structural and motor proteins. Structural proteins, for example, is this one here in the middle, which is collagen, which is, I think, 25 to 35% of the total protein amount found in the body. And they actually give our skin and our body and the connective tissue structure and flexibility and strength. And then we've also got motor proteins, which kind of work in a similar way in that they allow us to move. And one of them, for example, is myosin, which is responsible for muscle cell contractions and allows us to actually move and wave our arms, for example. Then we have transport and storage. We can make molecules with enzymes, but we also need to get them to where they need to go. A very classic example of a transport protein is hemoglobin in our blood, which transports the oxygen around and brings it to where it needs to go. Now, we don't want to produce protein in excess and waste resources if we don't need them. So we have proteins that specifically control the levels of the proteins in our body. And so depending on, like, if we eat food, regular proteins activates that enzymes are being produced to break that food down. And then once that is all digested, that food, we stop that process of making these enzymes until more food is ingested to just not waste resources. We also need to be able to defend ourselves against infections. And for that, we have, for example, antibodies and part of the defense protein system. Here we see, for example, antibodies, 3 of them bound to this protein in the middle. And so that can't cause us any harm. But also as part of that, there's proteins involved in wound healing, for example. And lastly, we want to be able to communicate with our surroundings. And for that, we have signaling and sensor proteins that allow us to, for example, see, feel, and touch. And then the signaling proteins then communicate this through our body to bring the information to where it needs to go. And for example, upregulate the production of an enzyme, for example. Now, all of this is encoded in the mRNA initially, from which it's translated into that protein. The mRNA itself is a short-lived copy of a gene, which is transcribed from the DNA. The DNA sits in the cellular nucleus, and because it is such a massive molecule and being really, really long, only the bits that we want to access are being transcribed into RNA, which then shuttles this information out into the cytosol, so in the larger area of the cell, so it can get translated into the protein that we want. And what is a gene? The gene itself is a small section of DNA that encodes for a specific protein, which obviously determines the biological function. And this code, this gene is encoded in nucleobases. And these nucleobases are just like a language that our body can read and thus produce the proteins. And this code actually consists of 4 nucleobases. And these are adenine, guanine, thymine, and cytosine in DNA, or thymine exchanged to uracil in RNA. And just like letters, just like an alphabet, we can read these, or a biological system can read these and know exactly from the exact sequence what the protein is and how it's being made. And so obviously the exact sequence of these nucleobases in the DNA determines the genetic code and thus the function of an individual. And we know how to manipulate this code. We know how to read this code. We know how to get from protein to the mRNA to the DNA and to actually access this code. And we can actually use this information and use DNA and RNA chemistry to study biology and treat disease. One very famous or somewhat infamous example of DNA chemistry in action is a very recent one, in fact, is the coronavirus vaccine. We can use what we know about DNA and RNA chemistry to figure out what the exact genetic sequence is of the spike protein, which is covering the virus. And by isolating this specific DNA sequence without the rest of the virus, which causes harm, we can then use this to study the protein in isolation, but also we can make the mRNA from it and then use the mRNA and hijack our own body's way of making protein to let our body make the spike protein, which is exactly what this vaccine does. And so what we do here is we teach our body how to defend ourselves against the foreign protein, which is recognized by our defense system, without having all of the other things that the virus has that would cause us harm. And so this is a very simple and very effective way of using DNA and RNA chemistry to do something very important, in this case, producing a vaccine. And in this case, these are produced by Pfizer and Moderna. And I know several other diseases are being investigated to have a very similar vaccine being produced that uses the mRNA of a specific protein that can be recognized. Now, this is all nice, but it's not what I do. This is very much on the consumer side of things. I personally work on a more fundamental level on DNA and RNA chemistry. Specifically, I'm working on chemically modifying DNA so we can control when it gets transcribed. And I do this by attaching molecules that allow it to be controlled with light. And so we can use this to control exactly when we produce protein. Now, you rightly ask, why is this useful? And I will give you 3 reasons why. These are the ones that drive me and drive the rest of the people I work with on why we do this. And point 1 is we can use this to recreate biology. As I already said, biology is incredibly complicated. There's a huge number of regulatory mechanisms involved in biology. And one thing we can do here is just strip that all down to a single bit of DNA, a single gene, and we can say now is when we want to activate it, and then we can study what happens downstream. And so it allows us to simplify biology down to the DNA level and to very simple triggers, in this case light, to figure out what is happening. Now we can go one step up and actually start incorporating more and more components, including a cell wall. And so we can make synthetic cells. We can then recreate cellular activity in the lab and start figuring out how do cells communicate, what is necessary for a cell to do a certain function. And then if we go even one further step up, we can then start working with these systems in real cells and start using them as controllable therapeutics. As you can imagine, being able to control which genes get made or not made allows us to make therapies and turn on and off certain genes that might be harmful or actually very beneficial when we want to. So we go all the way from simplifying biology down to the DNA level to actually being able to influence a real living biological system using light. And light is, for this, particularly good because it has, in my opinion, 5 very important features. It has incredible control. As you know, if you turn on a light switch, it's immediately on. If you turn a light switch again, the light's immediately off. We have very precise control in time over when the light gets applied. But also, as you know, with laser pointers, you can really pinpoint exactly where that light needs to go. In addition, biology actually has not that many systems that do interact with light. We have hemoglobin, which obviously we know is red. We've got our eyes so we can see, but there's not that many systems actually, and we can control for those. Light is also applied externally, so you don't need to give someone an injection, for example, to activate it, or you don't need surgery to do anything with it. So that actually is another benefit. We've got a very large number of colors to use. So from the UV light into like green and red light up into the near infrared. And we can use those in tandem to actually do different things. And also light can actually get into tissue quite well. If you've ever taken a torch and put it against your hand, you can actually see the light coming through at you. So this is actually a really, really nice way of getting a stimulus into a person or into just whatever system we're looking at, because light can actually just pass through. Now, the system I'm working with might be a bit intimidating initially, as the molecule is very large. However, if we just take a deep breath, and I'll talk you through the important bits and we can ignore the rest, it will work really well. So the idea is to modify a piece of DNA with a molecule, and that molecule silences the DNA activity by shielding it from its environment. And then when light is applied, that falls off and the DNA can be read again by the transcriptional machinery. The molecule I use for this is this monster. Don't worry, it is very simple if we break it down. It has 3 major components that we need to know about, and that's it. The first one is over here. This is thymine. This is the nucleobase that I use to attach the molecule to. And we have this tiny bit of linker so we can attach the molecule to it. Then we've got the molecule, which allows us to control it with light. Here in blue, you can see this part of the molecule breaks apart when blue light is applied. It applies here and it breaks here at this orange line. And then the last bit that we care about is this part here, which is just a molecular biology has evolved to bind to a certain protein. And this protein is called streptavidin, and it's very, very large. And that actually provides bulk and shields the DNA. Now, this is what it looks like. You've got these gigantic blobs of protein that really wrap the DNA around. And silence it and so can't fulfill its activity. And once we irradiate it with blue light, it breaks at this orange line here, and all of this massive bulk with the protein just flies off, and the DNA can do whatever DNA does again. And then we can actually use this. And one way we use this is in the control of synthetic cells. Now, a synthetic cell, as I said, is a compartment that is bound by a cellular membrane. And if we put all the things inside that are necessary to produce protein from DNA, we have basically a functional cell that mimic a real cell by the fact that it can do exactly the central dogma of biology. It can do transcription and translation. Now, if we put inside the DNA that I've modified, these synthetic cells become light activatable. And in a very simple experiment, I made these synthetic cells with a dye inside so we can see them, and then put my DNA inside as well so we can control them. And if we don't apply light, we only see the dye. The protein that should be made is M neon green, which is a bright green fluorescent protein. So once we radiate that with blue light, we're actually seeing that. We can see that the cells light up, they produce the protein, and that means we have now synthetic cells that can be controlled externally with blue light. We can go one step further, and we can immobilize the synthetic cells by placing them in a gel so that they move, and also express M neon green inside them. And doing that through a photomask allows us to pinpoint exactly which cells get activated. So I've used a dot photomask here where you can see very clearly 2 dots that have been patterned, and only where the light has hit the cells we can see expression of M neon green. So very much a clear boundary here for both of these where the light has hit them. And the same works with other patterns. So I've also used a line. We can also see a very nice straight line going through the cells, and only those have been illuminated and produce M neon green or whatever gene we want to put inside at a later time point. Now, this is all nice, but we can also do this to recreate biological systems, as I mentioned earlier. That becomes a bit more difficult because obviously now biological systems don't just produce one protein, they usually have more. And for that, I actually use a different molecule that my supervisor has made that is activated by UV light. And using that, I can have protein A being produced with blue light and protein B being produced with UV light. And then we can measure the interaction of protein A and protein B. Just like in a real biological system, proteins interact. And so we can make model systems that do very, very similar things. And we can measure the interaction of protein A and protein B. The system I have developed is kind of like a computer where you need both wavelengths for something to happen. Now, I've made the UV controlled and the blue controlled DNA, and they encode for 2 different proteins. These proteins are alpha and omega, and they obviously get activated or produced by irradiation with UV light or blue light. Now, when both of these, alpha and omega, are present, they assemble into an active enzyme, and that enzyme can then process a substrate. And the substrate, the small molecule that I've chosen here, is this carboxymboliferol beta-degalactopyranoside. Yes, I have practiced that. I call it CUG most of the time because the name is quite long. And all the enzyme does is it takes off this fragment here on the right. As you can see, this is obviously lacking this ring structure here. And this small molecule that remains is actually fluorescent, so we can measure it. And this actually is what is called an AND gate because we're having 2 inputs, UV and blue light, and only when both are present, we have an output, in this case, fluorescence. And this can be tabulated here. We see with 0 being no light and 1 being light. We see with no light, either UV or blue, we shouldn't see an output. With UV or blue, we shouldn't see an output, but with UV and blue, we should see an output. Now, I've done this in bulk in a test tube, and we can actually see this works. This is just a DNA that has never been modified, and you can see fluorescence. And if we don't put DNA inside, we don't see fluorescence. Now, if we put the modified DNA inside, we see also no fluorescence, following exactly what we see here, because we haven't illuminated sample yet. Once we illuminate with UV or blue, we see a tiny bit of fluorescence. That's because most biological systems are leaky and not perfect, but it is negligible in this case. And then once we actually apply both wavelengths, UV or blue, in either order, because it shouldn't matter, we see activity again that is very reminiscent of the DNA that has never been modified, meaning we have actually made a DNA logic gate, and also we've made a system that allows us to study interaction of two proteins, in this case on a model system, but that can be later used on a different one. And we again can put that in synthetic cells. In this case, I've immobilized them again in the agar gel and then used a photomask again to pattern them. And so we've got a vertical blue line and a horizontal UV line. And only where the lines cross, we should be able to actually express the enzyme again, because we've got both alpha and omega being expressed from the DNA parts. And then they can process, in this case, a slightly different substrate, but it has exactly the same properties in that it fluoresces afterwards. And we should be able to see that. And that's exactly what we observe, where only at the intersection of both of the lines, that this beautiful cross by UV and blue, we can see the synthetic cells light up, and actually we have enzymatic activity. And you can really see how tight this is by having a very, very low background and no activation anywhere else, except for a bit of noise, which is very normal. Now, we can activate genes. That's really good, but what else can we do? Now, there's also the part of translation that we can work on. And lucky for us, there is a mechanism of biology that allows short bits of DNA to interfere with this process via an enzyme. Now, being able to inhibit translation obviously is very, very important, because that allows us to, for example, stop a disease-causing protein from being produced, such as, for example, in Duchenne's muscular dystrophy, or in some cancers, we want to be able to stop a certain protein from being produced. Now, one thing that light offers in this case is that this makes this process controllable, because we can activate this short bit of DNA whenever we want. And because these short DNA sequences can stop production of protein and can be used to treat disease, being able to control them will improve how well they work as therapeutics. Currently, chemotherapy, for example, causes hair loss as a side effect because it goes everywhere in your body and does its effect everywhere. But being able to control it with light allows us to target a cancer specifically, for example. Now, this process actually is very interesting in that this bit of DNA can recognize an RNA that is very specific in sequence to the DNA. And that then gets recognized by this enzyme and cuts it in half. And so we've got two mRNA fragments, and they can actually not produce functional protein. The way we measure this degradation of the mRNA is via a process called gel electrophoresis, which separates bits of DNA or RNA by size. And we can see here, this is the full-length mRNA that we want to target. And in the absence of light, we see a single band here and nothing underneath. But once we illuminate with light, we actually activate this enzymatic process that cuts the mRNA, and we can now see two bits here corresponding to the two fragments that we've produced via the enzyme. One way I've been doing this in my PhD to control this process is by handcuffing the DNA. By looping around the DNA with the molecule that's synthesized and actually cuffing it to the protein with as if it were hands, we stop it from being able to bind to the mRNA, and that allows us to control when it works. And so obviously, again, when we illuminate it with blue light, this breaks off the protein and can do its thing. And actually works really well again. So this is the mRNA that we want to target. And this is just with the short bit of DNA here without the attachment of the protein. And you can see the two very clear bands of it being cut via the enzyme. Once we handcuff it to make this very big structure here, we don't see these bands anymore. And we can actually stop the process of happening, but we can recover that once we illuminate with blue light, and we can see the two bands again, meaning we're starting to degrade the mRNA, which will then stop the production of the protein. Now, here I've done this with one wavelength, but again, you'd be interested in, could we do this with two? Because that would allow us to generate a way of targeting different parts of the body with two different drugs simultaneously without affecting the other one, because we can activate them at different positions at a different time point. And for this, here I've made a UV activatable DNA that targets this lime green mRNA M. venus, and a blue light activatable DNA that can target this red M. cherry mRNA. Now, if we illuminate the UV one, we're going to cut the lime green RNA. If we illuminate this blue one with blue light, we're going to cut the red one and actually get 2 fragments again. And the nice thing is, because these bits of DNA are specific to the mRNA that we want to target, the other one should not be affected and we keep them intact. And that's exactly what we can do in a lab, where we can see M. venus and M. cherry. We've got them here, just how they're supposed to be. And then once we add both of these, unmodified, we can see very clearly the bands that correspond to them being cut. Now, once we modify the DNA, they are not getting cut anymore as we want. And then once we illuminate them with UV light, we're only activating this purple one here, and we're only cutting M. venus. Once we illuminate with blue light, we're only activating this blue one here, and we're only cutting M. cherry. This will in the future allow us to control which proteins get expressed, rather than from a DNA level on an mRNA level by stopping a certain protein from being produced, and actually allows us to make orthogonal therapeutics that allow us to target different genes at different time points with 2 different therapies at the same time. And this process also works in synthetic cells again, because that's what I like, that's what I do. And I again made them in this gel so they don't move. And I included M neon green DNA, which shouldn't be targeted by the blue DNA that I made, and M cherry, which is targeted by the blue DNA. So when we illuminate that through photomasks again, we're going to cut the M cherry RNA and we shouldn't see the red protein being produced. And how that visually shows in a microscope is that we're only seeing a green bit there because the red one isn't produced. And that's exactly what we observe with both the line again and the dots, where you can clearly see a zone that is only green because no red protein is being produced. You can see that here in the red channel of the microscope, where we see the trench and these dots. But the green channel seems to be completely unaffected by this process, again showing that we're not having any off-target effects using these therapeutics. Now, this is all a lot of data, I know, but what does this actually look like in practice when I do this in the lab? In one very short sentence, it looks very fun. It is very, very colorful. Obviously, these molecules interact with light, so they all have a color. So I've made compounds that are yellow, that are orange, that are pink, molecules that glow, molecules that are like a really deep, rich purple. We've got beautiful separation on column chromatography with these beautiful colored spectra. We can then crystallize our compounds out and they produce these beautiful yellow powders. And this is what gel electrophoresis looks like with my samples that are obviously also very colored. And so there's a lot of color involved. But because biology works on a very small level, all the tubes are tiny. So I've got these racks here that actually hold 96 tubes. And so I do a lot of things on very, very tiny volumes that are like tiny water droplets, basically. And one convenient thing is that the molecule that I work with does glow. So actually, I know where it is by just shining a black light at it. Now, to summarize, I have made a chemical tool, so a small molecule that can control DNA function with blue light. I was able to use this to make responsive biological systems from scratch by actually just making protein using blue light. And this can actually be then used to make DNA-based drugs that reduce off-target effects and actually target a very specific protein. And so there's a lot of potential for this work to be useful in both fundamental research in actually making these cells from scratch, but also in developing therapies from this. Now, a lot of this work wouldn't have been possible without assistance. There's everything from very hard chemistry all the way down to like deep biology. And so we have a whole team of people, my supervisor, Mike, as well as my co-PhD students, Jeff, Giacomo, and Razia, who really helped me along with this a lot. But also everyone else in both the Booth group and the Bailey group that I've been working with just for their continuous support, showing me how certain equipment works, or just going for a cup of tea when experiments don't work. Now, if you have any questions, please put them in the Q&A box, and I'm very happy to answer those.